So welcome everybody to the Human Nutrition Research Center on Aging at Tufts University's Monday seminar. Today is such a great honor, and we have as our speaker Dr. Patrick Stover from Texas A&M, where he serves as the Vice Chancellor and Dean for Agriculture and Life Science, as well as the Director of Texas A&M's AgriLife Research. Dr. Stover joined Texas A&M university system in 2018. He had been before then at Cornell University, where he was instrumental in growing the Cornell's nutritional sciences program through significantly increasing the research funding and really expanding its portfolio of academic programs. In 2016, Dr. Stover was elected as a member of the National Academy of Sciences, and he also served as the president of the American Society for Nutrition. He has achieved far too many other awards for me to name, and indeed I was tempted just to say this is Patrick, as you would all know him. But I want to add a few details about his biography to inspire many of our young investigators in the audience. In 1996, Patrick received the Presidential Early Career Award for Scientists and Engineers from President Clinton. This is the highest honor bestowed by the US government on outstanding scientists and engineers beginning their independent careers. Dr. Stover's research team focuses its work on studying one-carbon metabolism. The group investigates the chemical, biochemical, genetic, and epigenetic mechanisms and associated pathways with the one-carbon metabolic network. He also has worked, I know, with Joe Mason in our group. He recently conducted a very important workshop at NIH on the topic of one-carbon metabolism. But we invited Dr. Stover here today to present this lecture on his research and extensive knowledge and insights on precision nutrition. For those of us dedicated to advancing the importance of human nutrition, these are very, very exciting times. The NIH is promoting its Nutrition for Precision Health initiative with a significant investment and high expectations that new methods will be discovered for predicting individual responses to foods and dietary patterns. I recently heard Dr. Stover present his viewpoints on this at a USDA workshop, and I was so inspired, I invited Patrick to come and present, and I'm very, very grateful that he accepted. So it's time now for Dr. Stover to present his lecture. He will take questions after the lecture, so please use the Q&A box to send your questions. So with no further ado, we're going to hand over the screen to you, Patrick. And again, thank you very much for joining us today.
Thank you very much. It's a real pleasure to be with all of you here today. I'm here in Texas, where the weather actually is much better than it was last week, and we are grateful for that. But today I was asked to speak, about a little bit about nutrition research and directions based on, again, the talk that Dr. Booth heard me give, as well as to try to integrate that with my research. So you'll be hearing sort of a blended talk that hopefully will make some sense and will inform each other. But just want to say what an honor it is to be part of this seminar series. The HNRCA is world-renowned in what you do in nutrition and is certainly a model for all nutrition programs to aspire to. So I really appreciate the invitation. It's a pleasure to be here. So the title of the talk is Population Heterogeneity in Human Nutrition. Where do we go from here? Those are my disclosures. I have no financial interest in anything I'm going to present today. This is my lab group. This is the group that I had in Ithaca, who will have contributed to most of the work I'm going to present today. Most of them have moved on, but these are the people who actually do the work. We just take the credit for it, as you know. But I want to start out by talking about what seems to be the greatest challenge of our time, and certainly this century. And you're hearing so much noise about this, both in the media and within our funding agencies, is the real dire need to better connect food, people, economics, and the environment. We know each of these domains is really a complex dynamic system. And we all know that they all affect each other. Food affects people, food affects economics and the environment, et cetera. And we need to bring all of these four domains into greater harmony. And it is going to take a large effort, and it's going to require us to think differently about each of these domains and what the scientific evidence we need is to really affect the change we're all seeking. So if you look, the expectations of food and agriculture have never been greater. So if you look to the left there, that cover of Nature, Can Science Feed the World? And this was, again, relating to the growth of the population, the extra 2 billion people that are going to be on this earth over the next 30 years. And really, can we produce more? And that has been the historical expectation of agriculture, is to produce food, fiber, and fuel for the economy. But as you know now, the past decade or more, we've had these new expectations around nourishing and sustaining. Now we expect food and agriculture to promote health, to protect and sustain our environment so that our resources are there for future generations, and also to ensure that agriculture is economically viable. If food and agriculture is not economically viable for our producers, we're going to continue to lose precious land to other resources and lose that capacity to make food. And you actually see here in the Agri-Prop bill in 220, some of this being realized here, where there was, specific language saying the Human Nutrition Research, the committee that directs ARS, that's you guys, right? Provided the committee a report of how healthier food can enhance overall health, reduce obesity and comorbidity, and lower healthcare costs. So this is a huge challenge for us in the nutrition community now and in the agriculture community, as we are now, have this expectation of lowering healthcare costs that nobody can afford. And obviously this is because diet is a major driver of healthcare costs. Diet-related chronic disease is a major driver of healthcare costs. Of course, this expectation is not new, of really linking nutrition to lifelong health. And I love this quote from Jerry Fink, who was director of the Whitehead Institute at MIT during the Human Genome Project. And this was a quote from his presentation at the Lasker seminar back in 1998, where he said, "I expect in the year 2005, when the entire human genome is sequenced and mapped, the field of nutrition will be a completely different field. That will be the most revolutionized field in 2005. We will know a lot more. We will actually know something about nutrition, so you won't pick up one day and say fat is good for you, and the next day fat is bad for you, because we will know that for some people it is good and for some people it is bad. We will be able to know what it means to metabolize and what people can't metabolize. We're going to have a new definition of what it means to be healthy." So these were the expectations for 2005, resulting just to getting all the genomic information which we have now. And of course, the goal remains the same, right? To have a new definition of what it means to be healthy and how people differ in their response to diet. But clearly, genetics hasn't been the sole solution here. We now understand it's much more complex than that. We also see that we've really raised the bar and changed expectations of how we set normative guidance for nutrient-based recommendations. Where in the past, we really set those recommendations based on adequacy or to try to avoid deficiency. But increasingly over time, we've been moving to chronic disease endpoints. That is, if you're going to use food to lower healthcare costs, then you have to understand that connection between nutrients and chronic disease. And this was codified more or less in 2017 when the National Academies published a consensus report, a framework for establishing chronic disease endpoints when we derive DRIs. But of course, this is more aspirational than anything else, because as we know, few chronic diseases are not affected by a single nutrients in a single pathway. And that tends to be the way that we approach now setting endpoints for deficiency, for avoiding deficiency. We know that chronic diseases are complex with many interacting factors, and you have responders and non-responders. We know now that to achieve this, we have to consider systems and networks over pathways, because we know that chronic diseases are really systemic failures. We need to establish system readouts as biomarkers, integrative biomarkers. That is, biomarkers that really reflect how overall metabolism is working and how that then affects with systems related to chronic disease. We need to understand biomarkers of aging. One of your biggest risk factors of getting a diet-related chronic disease is to age. And so we have to understand the aging process and how our biological systems decay with age and how nutrition either accelerates that or decelerates. That's system decay that we know occurs. And so all of this has really accelerated the need for precision nutrition, where we're seeing that big emphasis now. And that was articulated both in the NIH Precision Nutrition strategic plan that was released this year, as well as you can see elements of that in the USDA Science Blueprint. So what is the why to precision nutrition? Why is there a sense that we have to go down this path? Well, again, if you look at diseases of nutrient deficiencies, they tend to have a single cause. You lack that one nutrient, and they manifest similarly in most populations. And if you're a geneticist, akin to what a monogenic trait would be. But diet-related chronic diseases are complex. They're like a complex trait in their etiology, with many interacting factors that exhibit great heterogeneity in populations. So it's akin to a gene or a polygenic by environment interaction. And so they are very complex. And this is why precision then is viewed as being needed if we really want to establish that strong connection between what people eat and what their lifelong health is going to be. We also see the why in animal models. And I'll just highlight what I thought was a really good study that was published in Genetics a couple of years ago by my colleague here, David Threadgill, who took 4 different genetic inbred mouse strains, 4 different strains shown here, and put them on 4 different diets and aged them out. So we put them on an American diet, we essentially just fed them a lot of junk, a Mediterranean diet, a ketogenic diet, and a Japanese diet. And they measured everything that you could measure: mutation rate, telomere lengths, all the classical biomarkers. And what they concluded at the end of this, other than the American diet, which wasn't good for any of the mice, you couldn't say that one diet was better than another diet. It depended on what your outcome of interest was and what mouse strain that you were looking at. So again, this model system in animals provides, again, evidence that we need to take a more precise approach to how we think about that connection between what people eat and their lifelong health. The how, of course, is a different question, because the science and the mandate are pretty clear. We've known for a long time that there are responders and non-responders and heterogeneity in that diet-disease relationship. But the pathway is not so clear. And that's what all the discussion now is about, both within our funding agencies and even within the popular press. And what I thought I would do now is pivot into the folic acid story, really to illustrate both why the concept of precision nutrition is needed, but also to illustrate why, or what the complications are going to be and the challenges are going to be in achieving precision nutrition. So just an overview of neural tube defects. This is one of the outcomes that we study in my laboratory, albeit in mice. And that is very early in pregnancy, even before a woman knows she's pregnant, generally between day 23 and 28, the neural tube closes. This is the head region, this is the trunk regions, the limbs will come from here. But during this time period, it's a period of rapid cell proliferation of the neural epithelium that's needed to close both the head and all the way down the trunk. If you don't have sufficient rates and coordination of proliferation, survival, differentiation, and migration of the neural epithelium during this time period, the neural tube will not close. It will remain open, giving you spina bifida here if you have opening here in the trunk region. And this mass you see here is not a tumor. It's cells that normally would have migrated down to innervate the legs here. Rather than doing that, they migrated out of the hole and formed a tissue mass. And this individual will be paralyzed from the site of the lesion down. Or you don't have closure in the head and you have anencephaly, which is always lethal. So what do we know about neural tube defects? They're quote unquote common rare birth defects. So they're a congenital abnormality in the US. This is the rate. Rate in developing countries is higher, telling you there's an environmental component. The recurrence rate, that is, a woman has one child with an affected NTD, then has another, is 1 to 5 in 100. And in fact, it does run in families. And that tells us there is a genetic component to this as well. And again, the critical period for closure occurs often before a woman even knows she's pregnant. And so that really makes any sort of an intervention difficult. But folic acid fortification to prevent neural tube defects has been one of the greatest public health success stories we have in nutrition. And rarely do you see the process work this well. Where in the 1960s, we had clinical observations that when a mother did present with a child with a neural tube defect, they showed biomarkers of folate deficiency. Now, not every mother who showed biomarkers of folate deficiency had an affected child, but those who did have an affected child more often than not had biomarkers of folate deficiency. That led to clinical trials, both for occurrence and recurrence of neural tube defects, which had to be stopped early because it was shown conclusively that when supplemental folic acid was given to these women prior to pregnancy, there was a 72% reduction in neural tube defect rates. Now note, this number is not 100%, so there were responders and non-responders, but the majority of the women did respond, and folic acid was the magic bullet. This led to recommendations: before you're pregnant, take folic acid. Of course, these recommendations aren't well adopted generally, especially with the target population here, which is young women. Then we go, so that then brought on a food policy where the FDA, under its authority to define what enriched means, expanded that definition to include folic acid at a particular level to prevent neural tube defects. And in fact, now we see in every country, in every place where they have fortified the food supply to lower rates of NTDs, they have all come down. The percent decline depends on what the rate was when you started, but it comes down to about 0.6 in 1,000. And it's believed that those residual neural tube defects are not folic acid responsive. They may be responsive to another nutrient, or they may not be responsive to nutrition. But this is an incredible success story where we have essentially eliminated in many countries this birth defect that was caused by insufficiency of folic acid in this target population. Yet if you look worldwide, a lot of countries still have not fortified. Dark blue is mandatory fortification, light blue is voluntary fortification by the industry, and white means no fortification. You see, many countries in Europe still do not fortify, which begs the question why. We have something, we know it works, we know it's effective, but not everyone is willing to fortify with folic acid for that outcome. And there are reasons for that, and I'll go through some of them. And again, this sentence here really relates back to, again, 1998, 2000. But this was the first fortification initiative that did not seek to remedy a nutritional deficiency. That is, even in the US, there wasn't widespread folate deficiency in the population. Rather, they sought a medical purpose, a clinical outcome, namely to remedy a rare disorder. So here you're using essentially a clinical endpoint for requirement. It exposes everyone, but only targets a small population subgroup. That is women of childbearing age who are genetically susceptible to having a child with a neural tube defect. So it's a small group. And this raised question of who benefits. And if it's a small group who's accruing a benefit, is there another small group who might be accruing risk? And what would that risk be about? And again, part of the reason of the uncertainty was we don't know the mechanism of how folate prevents neural tube defects. We still don't know that. This was sort of a black box. It was a clinical trial. You give folic acid, you prevent the NTD. We don't know how. But we also know that there's a folate cancer relationship. We know that low folate is a risk factor for certain cancers. And there is some thought out there that elevated or excess folic acid may also be a risk factor, but we still don't have the evidence base for that. Observational studies suggest that there is risk of high folate and low B12 status. Again, this is observational data. We don't have any mechanistic data around that. but again, is something that has to be investigated. Concerns about folic acid not being fully metabolized at certain doses, and maybe that has biological effects. But all of this creates uncertainty and concerns of unintended consequences, and hence not all countries have fortified. So I'm going to get more now into some of the research that we've done, trying to understand what are these mechanisms that can help then better predict the role of folic acid and what its role might be in multiple various clinical outcomes, and what it means at high dose and what it means at low dose. Well, what we know about folate responsive pathologies is there are many of them shown here: cancer, neural tube defects, cardiovascular disease, neurodegenerative disorders. And we know, though, that these really are complex traits, all of these. They are due to polygenic diet interactions. And that is, you need to have impaired folate status, either a primary deficiency or a genetic variant that keeps your folate status low, like the MTHFR gene, or mutations in some of the folate transporters. But this isn't enough. You also have to have impaired folate utilization. That is, you need a population that is sensitized to the deficiency. And you can be sensitized either through a secondary deficiency, such as vitamin B12 deficiency, or genetic variants, and we'll talk about some of those in the population that sensitize individuals to folate deficiency. So you need at least these two hits to give you the risk for these disorders. We also know that the outcomes, or the biology of folate and what it does in the cell, leads to even more complexity, because you need folate both to make methylation cofactors, as represented here by S-adenosylmethionine, which is a cofactor that's required for about 100 different methylation reactions, including methylation of DNA and chromatin, both histones and DNA. So folate deficiency is going to affect methionine and adenosylmethionine, and then have these genome-wide effects. But you also need folate, as we'll see, to make nucleotides. And if nucleotides are low, you have lower rates of cell division. You have elevated uracil and DNA that we'll talk about, and DNA instability. So you have these genome-wide effects. So getting to a mechanism between folate and a clinical outcome, you have to sort through the entire genome. And again, we really don't have that information yet. But the question we always ask in our lab is, is this the pathway? Is it all about methylation here? Is it all about me, or is it all about uracil and DNA, the U in DNA? And we still don't fundamentally have an answer to that question. Let me just speak a little bit about uracil and DNA for those of you who don't think about this every day. But the T base in DNA is the only base that is really non-essential. DNA polymerases, when there's an A on the template, will put a T during DNA synthesis on the newly synthesized strand. But if there is insufficient quantities of T, polymerases don't distinguish between T and U, and they will misincorporate uracil into DNA. So what you'll see here is that when thymidylate synthase, the enzyme that converts U to T, that activity is insufficient during folate deficiency, uracil will be incorporated. The amount of uracil that's incorporated is going to depend on this enzyme, DUTPase, which controls the level of DUTP available to be incorporated into DNA. And then you have base excision repair, where UNG1 is always scanning the genome, looking for uracils, chopping them out, and then putting the T base back in through DNA synthesis, repair synthesis. So you have a lot of players here that are regulating the concentration of uracil in DNA. U in DNA can have effects on promoter activities if it's in the right place, but also can lead to genome instability, because if you have too much repair going on throughout the genome, this will lead to strand breaks. So I want to quickly go over the one metabolic pathway and how we go about investigating this pathway. So it's really quite simple. It's three interconnected pathways, with the three products being purines, the A and G base in DNA, thymidine, the T base in DNA, and methylation reactions. That's the purpose of one carbon metabolism. It's found in three compartments. We'll see it's found in mitochondria, in the cytosol, and we'll see in a moment in the nucleus. But the purpose of mitochondria is to take serine and glycine. THF is tetrahydrofolate. There it is. That's folate. It's a metabolic cofactor that's going to really help extract a 1 carbon out of these 2 sources and release it as formate into the cytoplasm. Formate is then going to be condensed with tetrahydrofolate to form this activated 1 carbon group. So just as ATP activates phosphate for phosphorylation reactions, folate activates this 1 carbon for 1 carbon transfer reactions. This 1 carbon becomes the number 2 and number 8 carbon of the purine ring. Alternatively, you can pump electrons into that 1 carbon to get it to the 5-methyl state. Here you're going from formate to methanol to remethylate homocysteine to methionine that requires vitamin B12. That methionine is either going to be used for protein synthesis or other purposes, or made into a methylation cofactor for methylation reactions. Or alternatively, these 1 carbons can go to convert DUMP to DTMP to make the T base of DNA. Here, folate is both a source of a 1 carbon unit as well as 2 electrons. That tetrahydrofolate is converted to dihydrofolate. Dihydrofolate reductase then reconverts it to tetrahydrofolate, and then it gets recharged with a 1 carbon unit from either serine going to glycine through SHMT or through MTHFD1, formate ATP and NADPH going to 5,10-methylene tetrahydrofolate. Same reaction you're seeing here is shown here. And the reason I broke it out this way is the following. A fundamental question in our lab was, how do you make this decision whether to shunt 1 carbons for methylation or for thymidylate biosynthesis? We know that this is a really important decision in terms of maintaining both the methylation of DNA as well as allowing DNA replication. And how was that decision made? And so in studying that, we made a discovery that, in fact, these 3 enzymes here that constitute the thymidylate biosynthesis pathway are sumolated during the late G1, early S phase of the cell cycle. And SUMO is a polypeptide. It is a cousin of ubiquitin that designates proteins for import into the nucleus. And when they are sumolated, they go into the nucleus and they conduct 1 carbon metabolism in the nucleus. So while the purine bases are made in the cytosol, we have evidence now that a lot of the T base is actually made in the nucleus. So then one of the questions is, well, where are neural tube defects? Where do they happen within this network of 3 tightly linked metabolic pathways? Well, people have knocked out these enzymes here in the 1 carbon pathway that then restrict the synthesis of formate. And in fact, you do get neural tube defects. That's good news, but they're not folic acid responsive. So they're really not good models of what we see in humans. But they are formate responsive, which makes sense because to make these 3 products, you need both folate, but you also need the 1 carbon formate, right? But these really are a small percentage now, we know, of the human neural tube defects that are out there are resulting from mutations in these enzymes. Purines are not studied. This area, part of the homocysteine remethylation cycle and methylation reactions, have been studied all sorts of ways. These enzymes overexpressed, underexpressed. We've never seen an NTD, and when I say we, I mean the entire field, associated with physiological or pathological disruption of this pathway. Here, we do see neural tube defects, and we published the first folic acid responsive neural tube defect associated with a folate metabolic enzyme, SHMT1. So you knock this out and you see neural tube defects. And interestingly, you only need to knock out 1 copy of it. So it's highly, highly sensitive in terms of that neural tube defect readout. That is, you only need about a 50% reduction in the efficiency of this cycle to get a neural tube defect. And it's been known that methyltrexate and 5-fluorouracil, which target this pathway, also can result or increase risk for neural tube defects. So I want to go into a little bit of the biology of nuclear 1 carbon metabolism. Here you see the folate metabolic pathway for synthesizing thymidylate, the 3 enzymes. We took these enzymes and we made them or tagged them with fluorescent proteins. And so what you see for thymidylate biosynthesis here, if you express it in cells, you see in the nucleus here, it's present as fibers. It's present in the cytosol, but in the nucleus, it's in fibers. If you take SHMT1, you see the same thing. The nuclear filament is lamin B1. It gives the nucleus its architecture. It's also the site of DNA synthesis. And you merge all of these and you see, in fact, that this pathway exists as a complex attached to the nuclear laminin. And that attachment we found happens through SHNT1. This is the enzyme that when you knock it out gives you neural tube defects. It tightly adheres to the lamin and allows you to have synthesis of thymidylate in the nucleus at sites of DNA replication. And this just shows you how important SHNT1 is. If you look here, if you look at TYMS and how well that it co-locates with the lamin, you see very nice overlap. If you knock out SHMT here, SHMT1 and 2 in cells, TYMS is no longer localized to the lamin. It's all over the place. Hence, SHMT is critical for the architecture of this metabolic complex. If you knock it out, the complex breaks up. You see it here again with MTHFD1. I just want you to focus here, where if you look at a fluorescently labeled MTHFD1 protein, this is the enzyme that provides the 1 carbon. You see it's very nicely attached to lamin. When you knock out SHMT, it is no longer in the metabolic complex, and actually you don't get efficient thymidylate biosynthesis. We went on and purified that metabolic complex, and we in fact found the entire DNA replication machinery. So in fact, the T base is interesting because it is made on site, on demand, as part of the DNA replication machinery. And this is unique to mammals. You do not see this in yeast. In yeast, you do not have nuclear 1 carbon metabolism. It all happens in the cytosol, but in higher eukaryotes, it happens in the nucleus. So this just shows you the mouse. If you disrupt SHMT, again, just knock out 1 allele, you get exencephaly. There's the neural tube defect. This is a normal mouse, folate deficient, no neural tube defect. If you make a mouse folate deficient, you won't see a neural tube defect. You have to sensitize it genetically. By knocking out 1 copy of SHMT, you get the neural tube defect. And it's low penetrance. Only 10% of the SHMT heterozygote embryos get neural tube defects. So again, you see heterogeneity in response, if you will, that not all SHMT minus mice get neural tube defects. Only about 10% of them do. But if you give them higher levels of folic acid, they are all rescued, if you will. So it truly is a folic acid responsive neural tube defect. The only metabolic phenotype biomarker we see in these mice is elevated uracil and DNA. So if you look at uracil and DNA in liver in these mice, wild type mice, you have this much. You knock out 1 allele, it goes up about 6-fold. You get some rescue in the heterozygotes due to compensatory pathways that become upregulated. But that's the only phenotype we see is elevated uracil and DNA. Just want to point out here that we see other proliferative phenotypes. So if you look at neurogenesis, and we published this in the Journal of Nutrition about a decade ago, you also see impaired neurogenesis in these mice, the ones that are heterozygote for SHMT but didn't exhibit neural tube defects. So how does this relate to humans? Well, in fact, a couple of years ago, there was a study published that showed a human SHMT variant was a risk factor for neural tube defects. And this came out of case control and family-based triage approaches. And this is interesting. So I want to talk about this variant. This is the variant here. There's 2 alleles in the human population. There is a leucine and there's a phenylalanine. Leucine 174 sits on this helix. This helix is really important for UBC9 to dock to this protein, SHMT1. UBC9 is a sumo conjugase. So sumo, that polypeptide that designates this enzyme for nuclear import, sits right in here, and it modifies this lysine here with the polypeptide. When this becomes a phenylalanine, this whole helix moves back and interacts with this helix, and UBC9 cannot effectively dock to this protein. So this is the human variant that they're speaking to. In vitro, if you take SHMT and add ATP and sumo and UBC9, you can get sumolated SHMT. If you knock out these lysines, you don't get sumolated SHMT as you would expect. This is the modification site. With the polymorphism, you also get very low levels of sumolation. So this really points to nuclear one-carbon metabolism. You look at the mouse study, you look at the human variant studies and the structural biology, tells us that nuclear one-carbon metabolism is likely very important for neural tube defects. Just want to point out this is also of interest to NASA, and we've done some work with them, because a big issue in NASA is, as you know, disc edema and other ocular issues. Well, if you look at astronauts who carry that variant, this is the variant that gives you risk for neural tube defects, that impairs nuclear one-carbon metabolism, they're actually here at lower risk of disc edema relative to those who carry the wild type allele. So we're very interested working with NASA and trying to understand why that is. But what we know about human NTDs, from the clinical trials, we know that they're mostly folic acid responsive. We know that some are associated with an SHMT1 variant, low variable penetrance, risk associated with subtle alterations in metabolism. Also worth mentioning that kids with NTDs have a very high incidence of peripheral neuropathy and its early onset. If you look at the mouse model, you see very tight parallels. The NTDs are folic acid responsive. They also respond to DU, we found. Really a subtle genetic disruption, just one allele of SHMT knocked out. Low variable penetrance, again, of about 10%. Subtle alteration in metabolism. The only biomarker we have is elevated uracil and DNA. So we're going on to make other mouse models to really understand what is the relationship between SHMT1 and this neural tube defect phenotype. What is the mechanism? And for SHMT, we want to know, why are mice so sensitive to the expression of this enzyme with respect to the NTD outcome? Is it related to the catalytic activity? Is it related to whether that activity is in the nucleus or the cytoplasm? Or is it more related to its scaffold function and holding that complex together in the nucleus? Or is it something else? We also want to ask the question, does our mouse model exhibit peripheral neuropathies as seen in human spina bifida? So we'll go into these mouse studies that are ongoing now. So this is all unpublished data. But here you see DNA, here you see the beta-lamin, here you see the metabolic complex, the three enzymes that are making thymidylate with MTHFD1, converting DUMP to thymidylate. From here, DTYMK has to then carry that on to form DT. If this pathway isn't working efficiently, DUMP is going to be converted to DUTP and be misincorporated into DNA. So that's the pathway. And somewhere in all of this, we believe, is the neural tube defect mechanism. So we have we have about 5 mouse models actually where we've manipulated this pathway that are ongoing. I want to talk about 2 of them. One is we made an inactive SHMT, really asking the question, because remember, thymidylate here can get its 1 carbon from serine through this pathway or formate through this pathway. And in fact, most of the 1 carbons come from formate, not from serine, we know. So is it really the scaffold function that keeps this complex together, or is it the catalytic activity? So we expressed in mice a totally inactive SHMT1. I'll show you those. And then we wanted to ask the question, well, if this really is the neural tube defect phenotype, if we go 1 step down the pathway and knock out this enzyme, we should also get neural tube defects, but they shouldn't be folic acid responsive because this enzyme doesn't require folic acid, this pathway does. So I'll quickly show you the results from those studies. We made this inactive SHMT by mutating these 3 residues. This is folate in the active site of the enzyme. This is vitamin B6, which SHMT is a B6-dependent enzyme. Here you see glycine. These 3 residues here are critical for catalytic activity. And so we knocked out all of them. Here we substituted here Y for A, this tyrosine for phenylalanine, and this then K to Q mutation here. And this gave us an enzyme that folded perfectly fine, has the right structure, but is not catalytically active. Did they have NTDs? Yes. And in fact, they were more severe than just the knockout. So where we just got exencephaly when we knocked out SHMT, when we expressed this mutant protein, we actually got catastrophic craniorachescesias. That is, there was no closure along the entire neural tube. and we had a higher penetrance rate. May or may not be statistically significant, but they were very severe neural tube defects. So this tells us that having no protein is better than having mutated protein. It tells us that catalytic activity is important in addition to the scaffold function, and that when you express this inactive protein, it's actually stopping what probably are compensatory mechanisms. And so we're looking into that. We also knocked out this enzyme here, and in fact, we get beautiful high penetrance neural tube defects that are not folate responsive. So now we've walked up this pathway here. We continue to get neural tube defects, really telling us we're on the right track, that this is the pathway for neural tube defects that are folic acid responsive. Given that as we walk up, we continue to see the phenotype without the folic acid dependence. So we're going on to look at other ways of manipulating this pathway, really to get at the question, why do we get neural tube defects? Is it the presence of uracil and DNA in the DNA repair? Is it the slower rates of cell division because you can't make sufficient T and U is incorporated more slowly, et cetera? But we continue to look at this fundamental biology. I'm going to quickly go through now studies we've done to ask the question. Again, looking at the validity of this model in terms of human pathology. We know that humans, again, who get spina bifida have high frequency of peripheral neuropathy. What about these mice? Do we see that? And of course, we know that peripheral neuropathy is one of the symptoms of vitamin B12 deficiency. We know that if you are vitamin B12 deficient, you have megaloblastic anemia. Why? Because you impair de novo thymidylate biosynthesis. You trap folate cofactors as 5-methyl tetrahydrofolate. You can't make thymidylate very well. You can't make DNA very well, and you get these megaloblastic cells. Peripheral neuropathy is also associated with B12 deficiency, as is other neuropathies and cognitive disorders. So the question we wanted to ask again is this connected to our neural tube defect phenotype? And in fact, I'll just quickly flash this review in Nature a few years ago that really began to collect the evidence that DNA repair may be a unifying mechanism in neurodegenerative disorders. So we wanted to look at this two ways. We wanted to look and compare peripheral neuropathy in these mice, but also as a sort of a benchmark here, also look at diabetic peripheral neuropathies to compare both neuropathies that we might be getting in our neural tube defect model as well as what you see in diabetes. So this was the study design. We took the OB mouse that has diabetes, that's known to have an overactivated PARP pathway that's associated with DNA repair. And we have our mouse, the SHMT minus mouse with impaired 1-carbon metabolism, uracil misincorporation, DNA repair. And ask the question, do we see a peripheral neuropathy phenotype? And can diet rescue that phenotype? Again, this is all unpublished work. So these are the mice that we used. We did crosses with wild type mice that are heterozygous for SHMT. These are the ones that are the risk factor for neural tube defects. Our DB mouse that have diabetic obesity. And then we crossed the SHMT1 knockout onto that background, put them on a controlled diet or a folic acid deficient diet, left them on it for about a half a year. And then we looked at the phenotype. The phenotype we looked at was nerve conduction velocity. It is the gold standard for measuring peripheral neuropathies, where you have a stimulating electrode going down then to a recording electrode, and you measure nerve conduction velocity. If you have a healthy myelin, you're going to see a very healthy and optimal nerve conduction profile. If you have degeneration of the myelin, you're going to see a very abnormal nerve, and you're going to see an abnormal readout on nerve conduction velocity. These are the results. And again, this is unpublished, but this study is complete. So here we're looking at the sciatic motor nerve conduction velocity here as a function of genotype on 2 different diets. I'll tell you right now, it doesn't matter if you're deficient in B12 and folate for this model. We look at the control mice, this is the nerve conduction velocity. If we look here at the DB mouse, these are the diabetic mice that are known to get peripheral neuropathies, and we see a nice reduction in nerve conduction velocity. and that's highly statistically significant. If you look at our SHMT mice on a wild type DB background, so these aren't diabetic or obese, we see almost the same magnitude in reduction in nerve conduction velocity. So in fact, these mice not only get neural tube defects, are susceptible to that, they are susceptible to peripheral neuropathy. In fact, all the mice exhibit peripheral neuropathy. If we put both the DB background, the obese diabetic with the SHMT heterozygosity together, we don't see any magnitude of the effect, but we may be too late here. We have to look earlier to see if these two interact. So in fact, this model continues to reflect what we see in human NTDs, and that is in human NTDs, you see a high incidence of peripheral neuropathy, and we're seeing the same in these mice. So I want to now just go back to close, to go back to the original premise here of precision nutrition. And that is, how do we begin to understand and to do research around population heterogeneity in nutrition with the goal of lowering healthcare costs, lowering diet-related chronic disease. And again, folic acid is a model for that. We put folic acid in this food supply and we lowered healthcare costs, as spina bifida is extraordinarily expensive, and we have decreased dramatically rates of spina bifida in this country. But it really is a case study for the challenges. Number one, we know that those who are susceptible, have family history for neural tube defects, they are a sensitized population. So they are a subgroup within the population. If you make any mammal folate deficient, you don't see a neural tube defect outcome. You need to be sensitized to that outcome to see diet affect the neural tube defect rate. So you have a small sensitized subgroup that has a higher nutritional requirement with respect to a clinical outcome, and that is the birth defect. Second problem is we can't identify the sensitized group. There have been GWAS studies done on NTD cases throughout the world, and we still don't have a magic bullet. We have a lot of candidates. Some of them do relate to the pathway that I talked about, de novo thymidylate biosynthesis, but we still don't have definitive evidence from the GWAS studies as to being able to identify the sensitized groups. And as you can see, it's a very complicated pathway there in the nucleus. There's many genes, there's many factors. Not only do you have a small sensitized group, but within that group, you have responders and non-responders. The clinical trials, again, only rescued about 3/4 of the neural tube defect rate. So within the small sensitized group, you have responders and non-responders with respect to the folic acid intervention. And then you have the problem of the population-based intervention are not adopted everywhere. Europe is not fortifying with folic acid for the reasons that I gave. There are some people who are worried that when you benefit a small group, is there another group that may be accruing risk with elevated or excess folic acid? And Dr. Booth referenced this collaboration that Joel and I had leading this workshop at NIH about a year ago now, really trying to understand knowledge gaps and understanding effects of excess folate. Again, there are some observational studies. The literature is very mixed that there may be risk, but we have to understand this because we're benefiting a small group. Is there another group that is accruing risk? But again, you'll be able to look at this across the whole precision nutrition spectrum. That is, if you're going to see heterogeneity in response to that diet-disease relationship, and you take a precision approach, you have to then make sure that some groups aren't benefit, other groups aren't accruing risk. And again, the other thing to indicate, and that's really what I wanted to show you from the mouse studies, is the biology is really, really complex. When you talk about a clinical endpoint, you're talking about disease networks. And when you're talking about how do you look at these pathways and networks associated with disease, and then walk back then to the nutritional exposures through metabolism. You're talking about really complex systems biology and linking disease systems and then nutrition and metabolic systems. And that's going to be a major challenge for us. One of the approach that we've taken is we've teamed up with the Cosby group in Trento, Italy, which is beautiful there in the Italian Alps, but it's a Microsoft group that does hybrid stochastic modeling. So these aren't differential equation-based metabolic models. What these are is you start with an ordinary differential equation-based model, but then you lay over that simulations. These are very computationally intense, but you can then begin to look at entire networks and you can challenge them. So we have done this for the entire now folate metabolic network. We're behind in publishing a number of papers we have to get out. But we are now have a simulation of the entire one-carbon metabolism in the nucleus and the mitochondria and the cytoplasm. And we've done sensitivity analysis across gene expression for every single one of these enzymes, across mutations and polymorphisms in every enzyme, between varying substrates and cofactors. And we understand now what part of this system, what nodes are most sensitive to changes in gene expression or changes in metabolism, and really help show us where the sweet spots are in terms of the vulnerabilities of this network. And we can then go back and model those in our mice to see, in fact, if that's what we see in the biology. But these are the type of systems approaches that I think are going to be really, really critical in being able to really capture what is the opportunity for precision nutrition. So with that, I thank you for your time and attention, and I'm happy to take any questions.
Patrick, outstanding talk, and thank you so much for leveraging your vast expertise on folate and one-carbon metabolism to really illustrate the value of precision nutrition. So I'm going to ask the audience, if you have a question, please type it into the question and answer, and I will read it out to Patrick, and then he can answer. Joel, you has the honor of asking you the first question, Patrick. So Joel says, first of all, thank you for the terrific research and perspective on how you've integrated into the field of personalized nutrition. You see, Joel and I think alike. My question, if lower eukaryotes don't have internuclear DT synthesis, what are their compensatory mechanisms that enable them to have sufficient DT on site to synthesize DNA with high integrity?
Excellent. Excellent question. Thank you, Joel. And in fact, we have looked into that. We both modeled that in our computational model. That is, we took out nuclear one-carbon metabolism and just relied on the cytosol. And then we inputted all of the numbers from yeast, all of the expression level data that's out there and all of the activity data. And it turns out, if you don't synthesize de novo thymidylate in the nucleus through a complex, you need about 10-fold more of those enzymes in the de novo thymidylate biosynthesis pathway to make sufficient T bases for a round of replication. And if you look in yeast and you look at those enzymes, they have about 10-fold more enzyme expressed in the cytosol than what you see in a mammalian cell. So it is the de novo thymidylate biosynthesis cycle that is just upregulated in yeast relative to mammals. But then it does beg the question now, why did mammals decide to, or higher eukaryotes decide to move this into the nucleus? And that's sort of a fundamental question that sort of haunts us every day.
Great. So thank you, Patrick. So the next question comes from Alan Taylor, who also expressed accolades for your talk. And he has several questions about simulation. So the first question is, are people with simulation problems subject to neural tube deficits? The second question is, how many of the proteins that get simulated must be simulated to allow for nuclear localization? And finally, is there a competition between simulation and ubiquination, it is Alan, and degradation of the carbon metabolism machinery in the nucleus?
All good questions. So in mice, if you knock out sumo, you get neural tube defects, yes. So that's the answer to that. I am not aware of any polymorphisms in the sumo gene. Again, sumo is a short polypeptide that have been associated with risk. I will tell you a really exciting study we're doing. One of our colleagues at Scripps has been studying populations that have adapted to high altitude in the Andes. These people live in places where if you and I went there, we'd be dead within a day. And the question is, how do they adapt to that high altitude? But they also have a hematological profile that makes them look like they're B12 deficient, but they're not. And in fact, what we've seen, we're working with them, is they actually have polymorphisms that have a huge effect on the sumo-desumo pathway that directly affects de novo thymidylate biosynthesis. So there's something about that pathway that may also be related to, maybe not directly, to high altitude adaptation, but not neural tube defects, but we do see it in high altitude adaptation. In terms of the connection between ubiquination and simulation, absolutely. We published a paper on this in, I believe it was in Journal of Biological Chemistry. It may have been in PNAS a couple of years ago. I can send that to you, but there is competition. And essentially what we see in the nucleus is enzyme that doesn't end up being part of that complex. And SUMO is critical to form that complex in the nucleus. It gets desumulated and ubiquinated and then either degraded in the nucleus or it gets exported back to the cytoplasm. So there is a competition there between simulation and ubiquination and relates to whether or not you're in the complex or not. Absolutely. The middle question, I'm blanking on that, was.
Oh, sorry. Yeah, let me just, Alan was in the chat box, so I have to get back there. The third question is there a competition between simulation and ubiquination?
The second question.
Oh, I'm sorry. How many of the proteins that get simulated must be simulated to allow for nuclear localization?
Good question. We're looking at that from what we see right now. So SHMT is a tetramer. You seem to only need one. Only one monomer of the tetramer needs to be simulated for import. But we don't have definitive evidence for that, but that seems to be the case. What we do know that within that complex, not every subunit is simulated. What the purpose of simulation, it's needed both for nuclear import, but SUMO also is like the Velcro that helps multi-enzyme complexes stick together. So there's sites that are simulated, which I showed you, but there are also other sites on protein called SIMs, SUMO interacting motifs. And so they sort of are the Velcro that help SUMO connect two proteins together. And so right now we are modeling SUMO-SIM interactions and how SUMO is the Velcro that holds that complex together. But it seems right now that only one subunit is needed of the tetramer to exist within the complex.
Great, thank you. And Alan, thanks you for a wonderful talk again. Matthew Picklow says, very nice, elegant research. How can metabolomics be used to identify who will benefit from folate supplementation, i.e. linking the biochemistry and cell biology to predictive nutrition?
Yeah, we have done non-targeted metabolomics with our collaborator, Karsten Hiller in Germany. We have tried to look for all sorts of readily available biomarkers. The only one we see is genome instability. It's uracil and DNA, which is a real pain to measure, or an easier one to measure is just measures of genome instability. That is the only biomarker we see. We have done all sort of just broad-based agnostic profiling, and we do not see a metabolic signature that would, in our mice, where we know there's risk, that identify risk. It is all related to genome instability.
Great, thanks. Jiangdong Wang states, you mentioned that there is an insufficient body of evidence to support human adverse health outcomes that are a result of high amounts of folate or folic acid intake. Do you know if there is any study with upfront registration of studies of folate status and adverse effects?
Yeah, so I do not know of one. So there is an observational literature around levels of folate status or folate intake in a variety of outcomes, everything from autism to cancer to everything else. The data tend to be mixed, and given the study designs, they don't grade out very well. This doesn't mean that, obviously, if they're not important, it just means we need to get better data to understand, is there actually a risk or not? And then if there is a risk, then how do we take a more precise approach in understanding the dose-response relationship or the responder-non-responder relationship, so we can get the benefits of preventing birth defects and potentially other disorders without, others accruing risk. There's an interesting, we also don't have any mechanism of why higher levels of folate would be harmful. There's speculation. There's no sort of molecular biomarker that would indicate risk until Amanda McFarland just published in the Journal of Nutrition, I believe. I think it's available now. She showed a U-shaped curve for mutation rates in mice, dose response with folate. So if you looked at mutation rates, in deficiency, you have higher DNA mutation rates. Those then come down to a nadir. But then as you get to elevated levels, the mutation rate begins to go back up. We don't know why that would be, but this is sort of the only that I know biomarker, molecular biomarker that might indicate that there's risk. And those types of studies need to continue.
Great. It's after 1 p.m. and we have one more question. I'll just, it's a big question. So I'm just gonna tell people that if you need to leave, give them the time, we understand. So it's from Chris Wiley, who compliments your talk. And he said, since different forms of DNA damage can drive different types of cellular pathology, what type of DNA damage response is observed during neural tube defects, and is it nuclear DNA as opposed to mitochondrial that is primarily affected?
That's a good question. So we are looking into this right now. Let me just step back and saying it is all nuclear. So what's interesting about mitochondria, when I left Cornell, one of my senior research associates applied for a job at Cornell and got it. That's rare there. They tend not to take internal candidates. But we were looking at mitochondrial DNA, and she inherited that project. But what we did, and I believe she has published this, is shown that in folate deficiency, you see elevated uracil and nuclear DNA. Mitochondrial DNA is much more susceptible to uracil accumulation in folate deficiency. And so Martha is looking at that. She has a bunch of mouse models around that. But they are distinct pathways. That is, if you interfere with the machinery around nuclear DNA synthesis and metabolism and mitochondria, you don't see the connection there. You do see the connection with folate deficiency per se. And let me just point out another thing that's interesting. We did publish this. When you make cells folate deficient, the mitochondria will begin to become deficient, the cytosol will, the nucleus will not. We cannot make a nucleus folate deficient by depleting folate levels in animals or in cells. So the nucleus is very good at scavenging every little bit of folate that's there to keep it in that compartment. In terms of mechanism of DNA repair, one of the mouse models we have out there is UNG, and we were looking at the interaction of that with the neural tube defect phenotype there in the nuclear epithelium. So those are ongoing studies, and I don't have definitive data to tell you yet on that.
Great. Thank you, Patrick, for a wonderful, wonderful seminar. The engagement of the audience indicates just how riveting the talk was. Just one notice of housekeeping for our audience. Next week, we do not have a seminar, and our next seminar will be on March 8th. And it will be Danielle Belsky from Columbia, who is looking at biomarkers of biological aging using the calorie study, which of course was done at the HNRCA. Again, I want to thank our speaker, Dr. Patrick Stover, who's very busy, and we really appreciate you taking the time out today to talk to us. Thank you, everyone, and have a great week. Stay safe, stay warm, and stay healthy. Bye-bye.
Thank you all.